Hi, everyone. We are the mighty mitochondria. And today we will be answering the question of what is the difference between non-Mendelian inheritance and extensions from Mendelian ratio.
When talking about extensions to Mendel's laws, also referred to as extensions or deviations from Mendelian ratios, we refer to the genetic processes that differ in phenotypic expressions like codominance or incomplete dominance. These exceptions still follow Mendel's laws of segregation and independent assortment, but the phenotypic ratios differ from what is expected from a complete dominance relationship.
So if we look at a complete dominance, we see that we are crossing two heterozygotes. So what we're getting is the genotype of one completely dominant, one completely recessive, and two heterozygotes, which is a ratio of 1 to 2 to 1. Mind you, the phenotype here is a 3 to 1 of 3 depicting the dominant phenotype, which is blue, and 1 depicting the not so dominant phenotype, recessive is purple. When we look at incomplete dominance, the heterozygote is a blending of the two. So we see the red supposed to be fully dominant and the white supposed to be fully recessive, actually not being so totally dominant and presenting with an incomplete dominance pattern as seen in the heterozygote of the red and white blending to pink. Our genotype ratio, however, is still 1 to 2 to 1, but our phenotype has now changed to a 1 to 2 to 1, one red, two pink, one white. When we look at codominance, it's a little bit similar in terms of ratios because our genotype is still 1 to 2 to 1 following Mendel's laws, but our phenotype is now 1 dotted, which represents our supposedly dominant phenotype, and 1 striped, which is supposedly our recessive phenotype, but the heterozygote is a blending of both dots and stripes.
Non-Mendelian genetics. Extension from Mendelian ratio is not the same as non-Mendelian inheritance because non-Mendelian inheritance is the genetic inheritance that do not follow Mendel's law of segregation or independent assortment. These inheritances refer to a few cases which include epigenetics, maternal contribution, and a few cases of genome from organelles like the mitochondria or chloroplast that also do not follow Mendel's law.
In this image, it is depicted of epigenetics. In this image, we are shown genomic imprinting mechanisms where the methylation of an insulator located between the enhancer and promoter controls maternal imprinting of IGF-2. And in this image, we are shown that the maternal allele is silenced because of the insulator is unmethylated. In this image, we have mice showing the exposure to environmental stimulus, such as intergenerational versus transgenerational epigenetic inheritance. The color of the mice in this figure distinguishes different generations and do not denote